am Mrs. K. DEEPA, Assistant Professor, Department of Microbiology from AVS College of Arts and Science, Salem. Now I am going to take about Mycobacterium tuberculosis. Introduction. Mycobacteria are slender rods. They are sometimes show branched or filamentous forms of fungal myceliums. They are aerobic, non-motile, non-capsulated and non-sporing, usually living in the growth. They are acid force bacilli. Mycobacteria can be classified as Mycobacterium 1 and Mycobacterium 2. That means Mycobacterium tuberculosis and Mycobacterium bovis. The genus of Mycobacteria contains three groups. One is obligate parasites, opportunistic pathogens, next saprophytes. In obligate parasites, they include Mycobacterium tuberculosis complaints, Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium carami, Mycobacterium pindadi, Mycobacterium leprae. In opportunistic pathogens, they are the non-tuberculosis mycobacteria. They are contained a photomorphic under the gastrogenics of growers. Next, saprophytes. Saprophytes mycobacterium are isolated from many species. These included Mycobacterium from gases and Mycobacterium smagmatis from the segments. Mycobacterium tuberculosis and Mycobacterium bovis are human tuberculosis causing mycobacteria. They are discovered in Robert Koch. Morphology. Mycobacterium tuberculosis is a straight, slightly rod shape. It is 0.3 nanometer micrometer in size. They are occur in single or pairs in culture media. They are gram positive. They are filamentous. Next, they are in alcoholic forms. Cultural characters. The bacilli have slow growth with the generous. It contains maybe the slighter rods, and it have taken grow of 6 weeks. The optimum temperature is 37 degrees centigrade, and the growth not occurring below 25 degree centigrade to above 40 degrees centigrade. In optimum pH 6 to 7. Mycobacterium tuberculosis is a 0.5% glycerol improves the growth of the tuberculosis, but have not effect on the impact the growth of Mycobacterium bovis, whereas sodium nitrate helps us the growth of both types, Mycobacterium tuberculosis and Mycobacterium bovis. In several media, both solid and liquid media can be used for the growth of tubercle bacilli. Solid medium, the most widely used solid media as Lowenstein-Jensen medium. Next, on solid media, Mycobacterium tuberculosis forms a rough irregular colonies with the wide wall surface. Next, the colonies are creamy white becoming yellowish on puffy on cultural media. Next, what are other media used in the Mycobacterium tuberculosis are low-flux media and tertiary media, etc. In liquid media, the most liquid media used as the growth of tuberculosis. The growth begins at the bottom of the particles on the surface. Resistance. Mycobacteria are most completely heat resistant, being killed at the temperature for 15 minutes. Then culturals may be killed by exposure to the direct sunlight for few hours, but the bacteria survive in 20 to 30 hours and may be remain in droplets of 8 to 10 days. They are resistant to chemical disinfectants like glycerols, sulfuric acids and nitric acids or oxalic acids and sodium hydroxide in different percentage. They are sensitive to formaldehyde or glutaraldehyde. They can be destroyed by 80% of 80% of solutions in 2 minutes and by culture in 5 minutes. Host range. Mycobacterium tuberculosis causes natural infections in humans. It's a highly infectious to human beings. Mycobacterium bovis is more pathogens for animals. It causes tuberculosis in lungs and swine flus, other lung infections diseases. It is a mandatory pathogenic for humans. Antigenic properties. Whereas the antigenic has been identified in the pathogenic cell wall, they are consistence of lipids, proteins and polysaccharides. In lipids, the cell wall is present in the mycolic acid, where plays an important role in pathogenesis. Mycolic acids are also responsible for the cell walls of bacteria and cellular tissues in resistance to the bacterial bodies. Next, proteins, they are responsible for the tubercle secretions. Next, they included the hypersensitivity reactions, delayed hypersensitivity reaction. In polysaccharides, they have a pathogenesis on not affected. They observed in hypersensitivity. In biochemical reactions, niacin test. In niacin test, characterized by the addition of cyanogen bromide and the saline. Next, the reactions are positive infections means canons in yellow color debris. Next, Mycobacterium tuberculosis is the positive, Mycobacterium bovis is the negative. Next, anti-sulfuric test. In this bacilli grows in the medium containing 0.1% of tripotassium phenolic acids. Next, it have 2% NaOH added in the drop by drop. The positive reactions of pink color changed. Only the positive reactions are analytical measurements. Other than Mycobacterium bovis is a negative. Catalase peroxidase test. Equal volumes of hydrogen and ethanols in distilled waters are added into the 5% test culture and allowed to the strand. Then effectiveness for catalase positive. Browning is peroxidase positive. Already the analytical Mycobacterium candidas or catalase are positive and peroxidase are negative. In Mycobacterium tuberculosis, catalase are negative, then peroxidase are positive. These are the catalase peroxidase test results. In nitrite reduction test, sulfonamide and ethylene are added to the bacterial suspension in the nitrate medium. Next, positive reactions are red color. In Mycobacterium tuberculosis, red color is a positive and Mycobacterium tuberculosis, bovis in negative. Amidase test. In amidase methods, they are in 0.1% solution of the amide in the medium for bacterial suspension. And then magnesium sulfate and hydrogen peroxides are added into the phenolic solutions, and 0.5% of hydrochloride solutions are added. Then the amidase test reactions are blue color is the positive. Next, pyraminidase test. In this test, we have to conclude that pink color positive reactions is a pink color formation, and then Mycobacterium tuberculosis is the positive reaction, Mycobacterium bovis is the negative reactions.